All right. So let's go ahead and get started for today. So we only have just really quick. I want to go through the agenda for today with you guys. Maybe. And then after that, we're going to basically break into our groups and do our presentations. So first thing we're going to do, like I said, heroes and villains presentation. After that, we've got a lecture, which will be our last Venom lecture for the semester. And I did make a point because there's a lot of stuff I'm not going to get to cover. And when I checked yesterday online to see if you guys had questions about the poster, most people had not gotten their questions in yet. So we've got a big old section, hopefully, open for questions. And then the very last thing is I've got a little tiny survey I would ask you all to take before you take off for today. So before we get started with our presentations, any questions from you all? No. All right. Anyone want to volunteer to go first? All right, Athena, you are up. Look at that. You guys can come on up. Dr. Gangloff, do we want to share this screen as we're doing for the recording?
Yeah, I think we should.
Okay. Yeah. You guys come over. Which of these PowerPoints is you all's?
It should be shared. Oh, I don't know. What do we name it? Do you know?
Here, hang on. Here, hang on a second. I can help you out a little bit. Oh, I think it. Oh, is it there, or was it here? Or is it just because it probably is because I stopped the slideshow. So I would wait
It's
just a
that
second.
one.
No, it should still, it shouldn't do this even if you
Okay.
stop the slideshow. It should do something. It shouldn't be the blank blue screen. It should be just like the background of the desktop.
All righty. That's interesting. Oh, that was interesting.
I don't know what happened. It just decided that that should work. You might need to reshare the screen.
I will.
Yeah.
I'll have to do it between everyone, which is okay.
Oh, okay. Yeah. It should actually, no, I have it set up. If you set up just the share screen too, it should continue
kick it
sharing
back.
that screen even if you change PowerPoints.
Okay.
As long as you don't like unplug that, it should be okay.
Sure.
It's fine.
All right, floor is yours.
Oh, yeah. Okay, sorry that it's like kind of cutting off, but I'll fill that in. So we're looking at like the spiny bush viper and the scientific name Athenas hispida. And they're endemic to Central Africa. Primary habitat, they like rainforests, swamps, woodlands, anything kind of moist. Oh wait.
I can hit it if you want.
Oh,
Oops.
yeah, we can scoot.
Okay.
We can go to the side.
There you go.
Yeah, okay. I think I don't read this one. So they have a neurotoxic venom and there is no anti-venom for that. And their mating season is during the rainy season. Like I said, they like the moist habitats between the end of summer and October. They're semi-arboreal, so they spend most of their time up in trees hanging on. They also have like a prehensile tail to help them with that.
So this is our character, Athena the Athenas hispida, and her superpowers are that she can release her scales to hit targets, and using her pit organ, she puts out heat and makes fires. And her weakness is that if she falls out of the tree, she gets a scratch, and then when she has 3 scratches, she loses her superpower, and you can see that she already has a scratch on her face.
Okay, so how did Athena become a villain? So as a child, Athena's parents were up in a tree and the dry bush in Africa caught on fire and her parents were then killed in the fire while Athena helplessly watched. This is so bad. She couldn't leave the sight of the fire for days. And then she got sunburnt so badly that she got her superpowers. Athena now has a vengeance to burn all good creatures because she is filled with hatred. This is Athena.
All right, who's our second volunteer? Come on up. All right. What do we look for?
I don't know. It's like Venomous Hero or something like that.
Oh, if only we had a venomous hero one.
I think it's that one.
Okay, it's just
No,
the one right beneath.
yeah,
It?
that
All
one,
right,
yeah.
cool.
Yes,
Perfect. Perfect.
Yeah, yeah.
Great. Excellent.
Well, no, I'm right in the middle. I need to get off to the side. I'm too tall.
All right.
Okay. Who wants to speak? I can speak if you guys want.
Okay, yeah, sure.
Okay, our hero is Crateman. And Crateman is based on a banded sea crate. And their inspiration is, wait, what's that? Sorry. Oh, yeah. But their species is not particularly aggressive, but they have a super potent toxin enough to kill a lot of people with just one bite. And they can travel long distances and climb surprisingly high as well. And you can see their range there over in the red.
Mhm.
And
This is what Crateman looks like.
so he has the powers to, so he's obviously part crate. So he has the power to envenomate with the splinters from said crate or his crate arms. He has crate's four arms as well. He can grow human legs, but he's not human at all. It's all made of crates. And then of course he can float as well because he's a crate. And he can talk exclusively to crates.
Both the snake and the boxes.
Yes. And he could get them to do what he wants, like Aquaman, but he has to convince them first. So it's kind of like, hey, will you do this for me? So the secret identity is he's a floating crate with sea snakes drawn on the side.
You can kind of see here a little, just a box.
Yeah, so he just floats around the top of the ocean, just waiting to fight sea crime wherever it may happen. And of course, just like a crate, he gets, or a sea snake, he gets his water by drinking off the top surface layer after it rained. And the background was that he was a crate of smuggled crate eggs. And then they hatched and formed a brotherhood to fight against sea crime and those that abducted them.
All they knew was the inside of the crate, so they utilized that.
Yeah.
When they decided to fight sea crime.
And their weakness is large crowds due to their timid and shy nature. So if there's a large group of villains, they need to take off and go back to the ocean. And then we have a casting as well in the
We have
said
two
crate. Yeah,
actors.
two actors for one character in the said crate movie. So for the feet, only for the legs and feet, we have Timothy Chalamet.
He's in everything these days, so we
Yeah.
we have to take advantage of it.
And then for the voice, only the voice, it's Keanu Reeves. So it's like a Darth Vader type situation.
Yeah.
And yeah,
Yeah, that's it.
Thank you.
I ask this, does Keanu Reeves voice all the crates or just one of the crates?
I feel like they speak with one voice.
I don't know.
They like speak collectively.
Should be like one voice, but it has like a bit of like an echo effect.
We make Keanu Reeves record the lines multiple times and then
Okay,
lay
and
them
then
on
it
top of
overlays them. All right, all right. Man. And I guess I should have seen it. How was Crateman spelled? Is it C or K?
It depends on your reading.
Oh, okay. Sure. A local dialect. Great. All right. Who wants to follow up Crateman? All right. Chase, yours is superhero, right?
Yeah.
Excellent.
I have a creative title.
All
Can I
yours.
say something real quick?
Yeah, please. I want to make sure the slides are showing properly. I think I can adjust something. But it looks like. Let me just check. one
Oh, just wait.
I am.
Are there,
It's a
like, will the
snake.
speaker not show up there?
It's a snake.
If there are notes, no. Here we go. For that
Okay,
one.
yeah.
And these slides look okay, right?
There's
Okay.
And they're a
you'll
little
find
cut off, but
it's-
it's okay.
Is it? This one is?
Yeah, up there, but
Oh,
only the
shoot.
IA.
That's weird. Here, let me fix one thing, just because it's going to be a problem with all of them.
Okay.
Let me just fix it real quick here.
Oh, there we go.
Is that fixed?
Yep.
And that looks, okay, cool. Let me just go here. Let me just make sure. these look
Hold on to
okay.
go through the whole thing. Okay.
Yeah, yeah, I don't want to
There's
give it
this
too much.
part,
Okay, cool. Okay, yeah, sorry.
Okay, our superhero is Camellia Woman.
So, this is I think it's Capulus, I don't know, mud snake. So it was found, we think, in 2006. There's honestly not a lot of information on this species because it belongs to a family of snakes that is not studied very well at all. So this is what the snake actually looks like, I think. Like if you Google this snake, there's nothing. But they found it in 2006 and the scientists put it into a bucket and then she came back and it turned all white. There's no pictures anywhere of this snake actually being white. So these snakes are not it. Just like give you an idea of what it looks like. So it was discovered in Borneo. And like I said, there's not much known about the species, like why it changes color, because it was in the bucket. So there wasn't like an aggressor or anything that we know of. Yeah.
She's a mysterious woman.
Yes.
Not much is known about its feeding patterns, therefore not much is known about the venom either. It does have rear fangs, which is typically harmless to humans. She likes us, but can have harmful toxins in venom for other prey. And then
It delivers chemotoxic and, oh my gosh, I never can pronounce this. Proteolytic. Okay, cool. Venom to its prey.
So her superpowers, she has the ability to camouflage completely to her surroundings, which aids in the ability to sneak up or run away from other villains. She is a superhero, so she's gonna need that. She can shoot her venom to paralyze her villains. The rear fangs help with this because she's not like, you can't see them at first, and then she's like, "Papa, I can shoot my venom at you." So like they're little, they kind of hide it and not noticed by villains. She can shrink in size. These snakes are pretty small and kind of live underground, so that she can shrink in size to escape danger or sneak up on an opponent, kind of like the camouflage to her surroundings. Her weakness, though, is that there's not much known about her or her powers, so sometimes they get misused and she is unaware of her own powers, and then it ends up not being good for her. So when something new pops up, she's not aware of it, therefore she harms herself.
Self-destruction.
So her story, her first sighting was in Borneo, where she was found fighting with villains. They have only seen her a handful of times. She's very nice, but she can get mean. She only comes out when she's really feeling mean. Her camouflage abilities and ability to shrink make it hard to find her, obviously. And she doesn't like to fight villains out in public. Again, she's really nice. So she remains mysterious. And for our celebrity to play her in the movies, none other than Beyonce. Because women rule the world. And there's our sources.
impressive representation from the Homo-opsidae. Family which we do not know a lot about. All right, who is up next? All right. I give you guys all the credit. I believe it. All right, what are we looking for you guys?
Yeah. Yes, it's
Ah, I got you. No, why can't I? Wait, there it is. All right.
All right, so our supervillain is Sidestriker because we chose the stiletto snake.
And the original stiletto snake that we based this off of was a particular species called Branch's stiletto snake, or Actrasaspis branchi. It's native to the rainforests in Upper New Guinea, which includes Guinea and Liberia. The teeth of the Actrasaspis genus in general are known to be highly adaptive, so the morphology can change in a very short range of time in these species. But the Actrasaspis in general tend to have selenoglyphous fangs, so they were actually mistaken for vipers when they were first discovered, but they are actually colubrids. But their sister taxon, I'm gonna have a hard time saying this, Homoroslaps, have proteroglyphous fangs like elapids. So you can see that the fang morphology can kind of change very quickly in a short range of time. And of course, these guys are special because the fangs at the sides of their mouth allow them to strike sideways. So there's really no safe way to hold them.
All right, so the origin of our supervillain, it was the year 2011 in Liberia. Sidestriker was just an undergrad student tagging along with the research team, hunting down a new tree species belonging to the genus Apodicus. He found a stiletto snake and tried to pick it up because it looked super cool. It bit him since there is no safe way to hold a stiletto snake. He was given an experimental antivenom, which made the venom effects like swelling, blistering, and tissue damage spread really quickly through his arms. In order to save him, they cut his arms off. Little did they know that it was too late and his flexibility and paralytic powers had already spread. One day, he was presenting his research over conservation of his new tree species in Liberia, and a hater in the back yelled, "Trees aren't worth saving." Sidestriker glared at him out of the corner of his eye, and to his amazement, the rude man just stopped moving and tipped over, completely paralyzed. After that day, Sidestriker realized his powers and used them to paralyze those opposing conservation and save his newly discovered tree species. Unfortunately, authorities don't really like when you run around paralyzing people, and they often try to detain him. During one of their attempts to capture him, Sidestriker surprised Edelman himself by bending and coiling to elude the authorities. His super flexibility has allowed him to never be captured.
So as Judy said, you can see the powers are that he can paralyze people from just like a glance, because we were thinking these snakes can't be handled because they're able to bite with whatever like direction they turned. And then another power is that his extreme flexibility allows him to escape difficult situations, because snakes can coil their body around, and also he can't be held very well. One of his weaknesses is he did lose his arms, so his powers are primarily in his legs and his fighting style. And we have a. So would like to see
And between the illustrations and the celebrity representation, I don't know which I like better. They're all so good. All right, who's up next? Come on up. All right.
I just shared it with you. It should be like Sydney Simpson.
Okay, so
There's
it's just
email.
now.
Yeah.
All right, let me check
Under
here.
his-
Let's see.
We'll be fine. Got it.
Probably it.
I remember the song. I don't remember what reason I think I just loved it.
Got it.
I'm sure once I like the last thing. I figured I would just be left explaining my choices. Yeah, no, you're good, you're good, you're good. Yeah,
All
that's
right,
good.
cool, excellent.
Are you doing the information?
All right, take it away.
Okay. So, hi. We did the Gaboo viper. Its native range is the sub-Saharan Africa. And even though they have the biggest venom yield and the largest fangs out of any snake, it's not as deadly as we think. Unlike most vipers, when it strikes, it holds onto its prey for minutes at a time. Most vipers strike and then immediately detach their prey and follow them around. And despite the size of these snakes, they're often described as docile and rarely even strike.
Okay, hear me out. The super name's Gaboo because why not? You know, and it has they have super strength.
Great Gaboo.
The great Gaboo, exactly. It has super strength, scaly skin, and it can hypnotically make you calm because it's such a docile snake. And then so this is what happened.
Wait, no, you describe about its weakness.
All right, what is its weakness? Yeah, it's just totally uninterested in anything in life, in fighting crime. It's just the most bland-faced.
It doesn't
person.
care.
Yeah, it just
only
doesn't
does what
care.
it has to.
Exactly. Limited energy, guys. Limited resources. So they got caught up in the mob because they were just walking around and they were just chilling and they had that mean mug on and they got misrepresented for some guy. So the mob was like, yo, I know you're mean mugging me. We're going to take you out. And they was like, I don't know, man. I think I'm pretty chill. So they went and they got him to the desert because that's where they dumped their bodies, of course. And he was left there for dead, so he was alive when they left him there, and the sun just kept beating down on him, and so he got super dehydrated, caused him to have tough skin, and just by that sure power, he needed the willpower to have get really strong and fight these people off so that nothing else could take him away from his family and his life because he's running from the mob.
We chose a theme song for him as well, based off of his story of just trying to run away from the mob and stay alive. We figured, why wouldn't his theme song be "Staying Alive" by the Bee Gees? I can just imagine him walking to fight crime and staying alive in the background.
Oh yes,
it just works.
And then Gabbits, the Gabbites. Gabbits is his name, their name. It's very unisex. I feel like it's nice. That's it.
Right. Sam's up. I'm ready. Who am I looking for, Sam?
One of the many Sams.
Okay.
Which one is it called? It says Herp Hero / Villain. So
That is,
I have
I've seen
no
that one.
clue when that was shared with you.
I think it's probably this
We'll
one.
hope it
Let's
is.
try.
Probably.
Let's see.
Yes,
Oh,
that one
yeah.
is.
Perfect.
There's too
There
many
you go.
Sams here. All right, thank you. Okay, so our Herp is a villain to start off with. You wanna go?
So our inspiration came from a Cape cobra. Their scientific name is, don't know if it's Naha or Naja, Nivea. Native range is in southern Africa. a fact about their venom is that their venom is like very sticky, and it's also like really. They said that it was like thick, almost like maple syrup. And they can be found on farms, and they're extremely helpful in getting rid of rodent pests. But it's kind of strange because they're also like one of the most venomous snakes in Africa. So it's kind of a fun game for farmers.
So a couple more facts. It's a little blurry, but they have really like shiny scales. So his super persona is the Cape Halcyon, I think that's pronounced. Anyway, so his superpower is like because of like the shiny scales, we were like thinking like mirrors. So he is caped, obviously. So he has like a cape and he has like mirrors hidden in it so that he can blind like his opponents. And then because of his venom being like super sticky, we figured it'd be like Spider-Man and like he could like trap them like in his venom. And because of the mirror thing, unfortunately, his weakness is that if he sees himself in a mirror, he just kind of like gets captivated. He's like, oh my gosh, I'm so handsome. I'm like the greatest person in the world. So like he just kind of like forgets that the enemy's even there and just like, you know, he's just like staring at himself in the eyes. So yeah.
And this backstory.
The long
resembles
backstory.
This, yeah, it's a long backstory, so I'll kind of shorten it. Anyway, he's a narcissistic scientist working in South Africa where the snakes are at. He's working with their venom, trying to come up with a cure. He accidentally makes a really potent venom. He injects himself, but he got captivated in a mirror because he's walking by it, and he stabs himself with the venom. In order to find a cure, he rushes back and to the testing room and tries to find anything that would work, and he injects himself even more with a bunch of different random stuff. And instead of dying, he manages to become the snake essentially. His skin becomes pale yellow and there we go. Gains the abilities of Cape Cobra. However, his mind becomes warped and his narcissistic tendencies become even heightened.
And then
Oh,
he's
yeah,
played
Ryan
by Ryan Reynolds. So yeah. And that's it.
All right. What's our file name? What do you think?
Sandy, what is it called?
I think it's just superhero assignment.
That is a file name, so
You just look like
yeah, we're gonna go with that.
Oh, it's your.
All right.
Do you want to, which side?
Here you guys go.
Okay. Do you want to stay on this side? Or whatever you want, bro.
I'm staying here.
So I promise we didn't copy it, but we did in fact also do a stiletto snake, but ours is the healed huntress.
I'm sorry, I got anxious. I got scared.
These are also from sub-Saharan Africa and endemic to the continent. One fact about their venom is that the venom is potently cytotoxic, so it destroys the cell membrane, causing severe pain, blistering, and tissue damage. So maybe don't get bitten by them, but like that's your prerogative. And then something I thought was really cool when I was looking up about them is that they'll actually sometimes, because they burrow, so sometimes they'll actually go in and around termite mounds, because then they can eat anything that's preying on the termites. So they'll eat like lizards, rodents, and frogs.
Okay, so good old Brony. On a fateful day at a Foot Locker, trademark, he was trying on some discount stiletto heels, and little did he know, those heels were a little too tiny for his own good. A few steps around the store, he was vibing. Unfortunately, the shoes weren't either. So they got stuck, and about 40 minutes of panicking, he made a run for it and took the shoes with him. The next day, he awoke to a strange feeling, and the shoes had grown onto his feet, forming these permanent fangs shooting out of his Achilles. I don't know if that's actually how you pronounce that. Its fangs contained a venom so strong that it could capacitate even the strongest of enemies, and this was his origin.
So his weakness is the candle section at TJ Maxx, because he has really bad eyesight, but their scent is really strong. So just one inhale of that TJ Maxx candle section really breaks down his venom. But his powers are that he has venom also from his Achilles fangs, and also curb stomping. His celebrity casting would be Ben Affleck because we feel like he needs another chance after Batman's flop. That was all.
All right, what presentation am I looking for? That's a good question. Oh God. Here, hang on one second. I can help a little bit. All right, so who turned it in? Well, so hero slash is in this guy. Hero slash villain herpetology. Ooh, let me make sure I got that one. I think it's this one then. Yep, there we go. All yours.
You want to say it?
Am I going?
Yes.
So this is our villain. That's his name, the Killer Jaws. And it's based off a Gila monster. And right here is a fantastic idea of turning Gila monsters into wrenches. I'm just kidding. I got this picture off of Wild Kratts and found it funny. So I have it up there.
So some of the background information is that it is native to the southwest US and Mexico. The new fact is the Gila monster's venom. There's a part of it called Exidin 4, which is a gluconic-like peptide 1 receptor. When it's isolated from the venom, it is currently on the market to treat type 2 diabetes in Bayetta. The natural history fact that I found interesting was that I didn't know that there was 2 different subspecies of Gila monsters. The most well-known is the Arizona or reticulate Gila monster. It's so named because the patterns, the adults form a network of irregular black blotches on a lighter background. And then where's the other one? The other one is called the banded Gila monster. It is found in the Mojave Desert and the arid lower Colorado River subdivision of the Sonoran Desert.
Yeah, powers. Her first power is Venom Punch because we learned that Gila monsters, when they bite people, it doesn't kill them, but it hurts a lot. So the Venom Punch is when she punches people, it hurts like 20 times more than a regular punch would. And her other power is Unyielding Grip. Gila monsters have this tendency that when they bite, they don't like to let go. So when she grabs you, there's no way to escape. When she grabs you, you're a goner. And yeah. Weakness is she's super slow because Gila monsters are super slow.
We got the background, the identity is Hilo Sonora, and then the origin story. While they were out hiking one night, she accidentally stepped on a tail of a Gila monster. It then got angry and bit her, so she was stumbling around from the pain. And because she was stumbling around and walking around, she got bit by others, and then more, and then became very delirious from all the pain that her alter ego was born. She takes out any of the anger on innocent passerbys that she can get her unyielding grip on.
And there we go, works cited.
All right. All right, which file?
It's gonna be whichever one has Sicilian in the title.
That would make sense. There you go.
Sorry.
Maybe lost her presentation is.
For just
This is just for the notes. We'll click on the his laptop.
Yeah.
Yeah, she's named after the Alaskan bullworm.
Yes.
Ours is a villain. Her name is Dr. Alaska Bull, or secret identity name, or I guess villain name the blind one, because I learned that that is where the name Sicilian comes from. It's Latin.
So we actually have two species that we based our character off of. One of them is the ringed Sicilian, which this will be our main species and where our villain gets most of their powers. Their primary range is in the tropical South American rainforests. So it can mostly in like Brazil, but can range all the way down through Peru even. As we know, Sicilians are one of like the most understudied invertebrates, but it has recently been found that they do have venom. They were found to have toxin producing dental glands like a snake. And they were quite possibly the oldest known venomous creature on Earth. They possess an A2 phosphorylase, oh my gosh, I can't say it.
Okay, it's
I can't say it. Anyway, it's an
A2
enzyme.
phospholipase.
Yeah, it's an enzyme and it's in their saliva. It's the same as what's in a lot of arthropods venom, like wasps and scorpions, and as well as some snakes. And they have their glands, they'll work kind of like the Gila monster, where their venom will come down like as they crunch and munch on something. It just kind of coats their teeth. And they're very elusive and spend most of their time underground, and pretty much the only way to find them is by digging them up. Oh, no, I need to go back. I thought I had this clicked on so I could scroll through the notes. Oh, no, I was looking at my notes. Yeah, sorry. Oh yeah, they're also jaw feeders. So as you can see, they have some wicked looking teeth. They use those to latch onto their prey, and they have a very powerful bite for their size. And then our second species, we're using the axolotl because they're a pretty good example of a pedomorphic amphibian, and they grow back their limbs.
Okay, so now for the backstory. Dr. Bull began as a well-meaning, albeit a little bit odd scientist with a passion for amphibians. Her primary species of study was the ringed Sicilian, because she was interested in their mysterious venom that nobody seems to know that much about. And they also studied axolotls as like a side project because they were interested in their regenerative abilities. She struggled to maintain her lab financially, going off of measly little grants and her own personal funds. And then it was revealed to her by her landlord that the property was being bought out by an entrepreneur who planned to adopt the complex into overpriced apartments and rent them out. So she was going to be losing her laboratory. She worked alone, and given that the grants she received were very small, she couldn't afford to essentially say, hey, I'll buy this from this very rich person. And given that she is also a solitary scientist, tinged with just a tad of madness, she wasn't really that much of a fan of an authority figure kind of taking over her life's passion. So she developed a serum to give her an upper hand on the system. Had a bit of a taste for unhinged vigilante justice here. And they initially intended to use this to keep people in power from like walking all over people like her. And mysteriously, the entrepreneur planning out to buy out the property disappeared. And the complex remained as it was. And as they went under more transformations, they began to fight more viciously, started using more venom than necessary for incapacitation, and they relished their new power. And so it began.
So here's an artist's rendering of how she looks in human form. Just a very extra villainy outfit. It's just kind of like a messed up lab coat, and then this spooky foreshadowing shadow in the background. They just kind of always look tired, you know, just like done with everybody.
Like the overworked and underfunded scientist kind of trope.
And so, as we've mentioned, her powers are inspired by both Sicilians and pedomorphic salamanders.
Okay, so starting with the transformation, using her serum, she gained the ability to transform into a giant, horrifying Sicilian. During this transformation process, her arms and legs fall off, and she turns into this giant, scary, wormy amphibian. The weakness in this form is that they're blind, they can't see, because Sicilians are primarily subterranean dwelling creatures, and their eyes don't work very well.
So she also has little Sicilian minions that she started using from her like lab friends that she would get from the wild. And they are able to be summoned from underground as sort of just like angry little friends to help her out in the fight. They will bite off of little chunks of her to gain more energy, more power, and then they will literally swarm the enemy that they're trying to fight. Because baby Sicilians, as we saw, eat their mother's flesh. So that's how it works with her. So, King, did you want to talk about the venom? Okay. Her teeth are coated in venom. Like we said, it's just kind of a passive like chewing system. So the minions will just kind of latch on, incapacitate, and just kind of munch. And she's also mildly poisonous, as some amphibians are, poison dart frogs. She can burrow into the ground as long as it's not like rock. So if it's like asphalt, she can't, but if it's like dirt, she's fine. And then also one weakness is that she has to go through a recovery period, because once again, when the baby Sicilians eat their mom's flesh, she has to kind of wait a couple days to regrow it. So one, she has to do that, and two, she has to regrow her limbs. So she, after a fight, she's pretty helpless for a little while.
And this is her in her Sicilian form. Very, very scary.
That's all.
Yeah, and then we're excited.
All right. Is that it? Anyone else? I think that's it, right? All right. Well, first off, let's get another round of applause for everyone. That was exceptional. All right. And for the rest of us, we're actually just going to go ahead and take our stretch break at this point. So take your time. If you need to run to the restroom or anything like that, go for it. Check your phones. And when we get back, we'll do our last little bit of the Venom section.
Exit ticket with me.
Yes, I can.
Yeah.
Yes. Let me actually pull it up on my computer.
Okay.
So, and I will bring it over to you.
Okay.
All right, so without much further ado, I once again want to say thank you everyone for your heroes and villains presentations. I personally really enjoyed those. I hope you all enjoyed them a little bit as well. And we're going to now go ahead and talk for the last time about Venom, and we're going to cover a couple different things. The first thing I want to talk about is finish talking about the evolution of Venom as a phenotype. And so in that regard, the first thing I want to do actually is just cover a couple of topics that we've already discussed. So remember, and this is now something we really want to emphasize, Venom is this mixture of toxins. And we see that these are enzymes produced by the organism. Each of these enzymes have different functions, different compositions, have different functional effects. You guys have seen this slide before, so this is just a reminder. And we also talked last time specifically about the mechanisms that underlie these different sources of variation. And we know that the phenotype is ultimately, and at least for Venom, is controlled by the available toxins, the toxins that are in the venom, and the relative quantity, which is basically how highly expressed the genes are. And if we're talking about mechanisms of evolution, we identify gene-based mechanisms of evolution, which are duplications, and sequence evolution, and we talked about regulatory evolution. And so today I want to just quickly go over this gene and duplication-based evolution again, but really what I want to talk about is sequence-based evolution today, so that we can totally complete the picture, because this is not only indicative of how venoms evolve, but really more broadly about how phenotypes evolve, and venoms just provide a really cool example. So something we talked about last time that I want to remind everyone of is that gene duplication and loss is absolutely what's responsible for the neurotoxic and hemotoxic dichotomy we observe in Mojave rattlesnakes. So remember that Mojave rattlesnakes, the ones that have the neurotoxic venom, have two genes that encode a very potent neurotoxin called Mojave Toxin, and that our type B venoms do not have those genes. And we identified gene content, or what genes are present, as this very important axis of duplication. And we looked at this little graphic, which showed us the chunk of DNA that these genes sit on. And we saw that in our neurotoxic or type A rattlesnakes, we have these two neurotoxic genes. And here I've labeled them as subunit B, subunit A. And then our species that don't have the neurotoxin have two totally different genes there. So back broadly speaking to gene duplication and gene evolution. So one of the questions that we have is, if gene duplications occur, when they originally occur, it's important to remember that these genes are probably identical. So at the very first instance that a gene duplication arises. And this is kind of interesting because you might imagine that this could, in some cases, not change the phenotype very much. So there is this question of, how is this advantageous? Well, in fact, there are two ways it changes the phenotype. And the first is this. One possible advantage to having a gene duplication, this is especially relevant to venom, is that you increase expression, or you can increase expression. And the reason for this is that if we have a gene, like here I've got gene 1 and it's expressed a certain amount, many times when a duplication occurs, the regulatory elements are duplicated with it, or it might occur what's called in tandem. So it will occur right after the previous gene. And so you basically get this automatic increase in expression. So you go from having 5 expressed transcripts here, now you suddenly got 10, all just because the gene is duplicated. Now, importantly, in many biological systems, this is actually problematic because you can basically be producing too much of a protein. But sometimes in some venom systems, this could be advantageous. So for instance, imagine that you're a snake and you're chomping on something like squirrels or frogs or mice, and you've got this toxin that's really effective against them, but you don't have that much of it. One thing that might be selected for would be if you have a gene duplication of that toxin, you've now doubled the amount of that toxin in your venom just through that single mechanism, and that could be beneficial. So it's actually been proposed, this is a really important idea, it's been proposed that expression may actually be a key selective pressure favoring gene duplication in venoms. And we don't have, there's actually been a few different recent works that have suggested this exact same idea. And it's one of the things that we think might be responsible for why we see so many copies of these kind of similar venom genes. Now, once you have a duplication, there's another advantage or possible advantage of having the duplicates. And that's that once you have a spare, you actually have a relaxation of selection pressure to preserve gene sequence. So if you're wondering what that means, you got to think about the fact that proteins are very fragile in general. Small changes can have big impacts on their function. And in fact, the vast majority of mutations that are going to occur in a protein are far more likely to basically break it. So most mutations that occur in general are not good for your fitness. They result in a malformed protein or a less effective protein. And so most of the time, selection is actually trying to just preserve what you have. You can think of it as, if it ain't broke, don't fix it. You want to maintain it just how it is. But if you have a duplicate, so now you've got two copies, you become far less concerned about this constraint because you've always got a second one in the background. So you might think of it as a lot of different things. One example might be like a phone. So I'm sure everyone here has a phone. Who takes really good care of their phone? I do, or I try to. If you don't, okay, I can't really help you there. But normally for most of us, if we only have one of something, or another example actually might be your computer. A lot of us want to preserve these items that we have that are very important to us. But if you had two phones or three phones or even three computers, how much effort are you going to put into maintaining it when you know you've got another that's always available? It's essentially the same idea. By having a copy, you're much more robust to the idea of having one that's less effective because you've always got the other as a backup. So in this way, selection has basically been loosened. And I'm putting it here, it's because it has a spare copy of the gene. It basically means you have one that can maintain the original function. And that opens a lot of avenues for the second gene, especially with regard to sequence evolution. Yes, Sydney.
Can you give us an example? Because the only thing I'm thinking of is the second X-link or the second X chromosome that females have.
I am going to give a what I think is a super exciting example of this. So in fact, that's basically the next thing I want to talk about is I want to talk about a very practical example of how this all occurs. And this is actually going to be a case where really all three of these mechanisms come together, but it's especially a function of gene duplication and sequence evolution. So recall that toxins gain new or improved function through sequence evolution. The only way your toxin will change function is by a change in its underlying gene sequence. Now, again, in most of those cases, those changes are actually not good things, but sometimes they're neutral. They might not actually affect the protein. And sometimes they, in very, very rare instances, they can actually be advantageous and they would be selected for. And again, we know that this is more common when you have a spare copy. And this is basically true of all genes. And this is something that we see in Venoms broadly, is that we have a lot of these what are basically duplicated genes that have changed. And again, because of that, in Venoms, we actually see that these toxins and their sequences are evolving especially fast compared to other systems. And so on the right, what you're looking at here is just an example of a phylogeny that would be basically the history of a given gene or toxin. So here it's called here ancestor 0. And early there's this duplication. So there's now these two copies of the gene and they actually can both change a little bit and they start to do different things. Each of those copies then might duplicate again, or they might just through time have small changes in their sequence that after a while, and when I say a while, we're talking about a couple million years probably, we actually have very different toxins that we're dealing with. And so there's some really interesting cases of this occurring, but the one that I want to talk about with you all is a toxin that we've now danced around quite a bit. And I haven't given much attention to it because we're talking about venoms broadly, but I think this is a fantastic example. So this is Mojave Toxin. So we talked about neurotoxic Mojave rattlesnakes. We talked about how there are differences in these genes. Let's talk about what those genes actually are right now. So Mojave Toxin is this very potent neurotoxin. It consists of two subunits that work together. We have an acidic subunit. We have a basic subunit. Those are also referred to as the alpha and beta subunits. And if you'll remember from our picture a couple times ago, we actually saw these two genes. There were those two red arrows. So each of those subunits is encoded by a very specific gene. Now, importantly, in this toxin, our basic subunit is actually what does all of the work. And when I say all of the work, I mean almost all the work, because it is a neurotoxic toxin. So if you inject it into a mouse, it will have function and it will disrupt motor function, but only like at a sort of moderate degree. The acidic subunit, in contrast, its only job is to serve as a chaperone protein. And so actually, if you, for instance, inject the acidic subunit, yes, will. Yep, I will. I'm going to hop back, but I want to finish this thought really quick. So if you inject the acidic subunit alone, it has no effect. It's completely non-toxic. It doesn't do anything. It's only in combination with the beta subunit that it becomes 10 times, or it makes the beta subunit 10 times more toxic. I'm going to hop back for a couple minutes so you guys can finish copying notes here. Good. All right. Hop forward again. So again, the acidic subunit basically non-toxic on its own, but it makes the beta subunit much more effective. And then one last thing that makes this whole scenario much more complicated is that the acidic subunit has to undergo this very specific set of cleavages so that it will fit with the beta subunit. When I say fit with it, we're talking sort of like a Cinderella slipper type thing. You have to have just the right shape, and it needs to be just right so that it'll bind to the beta subunit enough to block everything else. But when it gets to the neuroreceptor, it lets go. Now, if all of this sounds complicated, it's because it kind of is. And in fact, I remember learning about this when I was an undergrad and being really baffled by the situation. Because one of the things that seems problematic about this is basically this question of, well, if the acidic subunit's only role is to assist the beta subunit, how did it evolve at all? Because there doesn't seem to be a selective pressure to maintain a toxin, I say that with quotes, that doesn't have a toxic function. And so that presents kind of this odd scenario that requires explanation. How did this change in sequences occur that allows it to be cleaved and allows it to bind to the beta subunit, you know, in a way that was not a huge selective disadvantage to the snake? And so just as a quick graphical representation of what's going on here, because I know that I basically verbally explained a very complex scenario, this is kind of what we're looking at. So here our gene 1 is representing our beta subunit of the toxin. And it's going to produce a product 1. So here, this is our nice little neurotoxin, does its own thing. But gene 2, it has to produce a product. And then that product actually has to be cut up by an external enzyme. So it's something completely unrelated to this system. And then only then is it going to be effective to fit with the actual toxin. And so we get this toxin complex. So again, our question really here is, how does this occur in an evolutionary framework? So how the heck did this subunit even evolve? Well, it turns out, and this is really cool, I think this is really cool. It turns out that all of this occurs through basically a single point mutation in the acidic subunit, or in what was the ancestor of the acidic subunit. And the way it works is this. Under normal circumstances, there are older basically variants of this alpha subunit that have similar sequence, but they've maintained their enzymatic function. So they're basically toxins that still work. But there's this one mutation that'll occur in the sequence. And here I've highlighted it. And I know that these letters don't really mean anything to you guys, but for example, these are our amino acid sequences. And these other lines are examples of other toxins. And you'll notice that in our very top line, which is representing the ancestor of all of our neurotoxic alpha subunits, these are our guys that need to get cleaved. We have this mutation here that produces a different amino acid from this proline that we see in these other toxins, these other PLA2s. Turns out that this proline is super important to maintaining the structure of that protein, because if you have a mutation, as we see here, suddenly there's a cleavage site that's accessible. And so if you haven't taken biochem or you're not familiar with this, This is the idea that other enzymes can actually bind to a specific point on the protein and basically cut it. And so in doing so, they change the conformation. And specifically, what's cool about this is that the protein that actually can bind to that cleavage site is another venom component. So we're talking here about a group of toxins called the PLA2s. These are phospholipases. You actually heard about this in your superhero presentations. But there's another group of proteins called the serine proteases. And in fact, they can bind to that site and chop it. Now, once that occurs, suddenly you get this big change in the shape of the protein, all from one point mutation. And that change opens up four more cleavage sites, all of which can be cut with other venom proteins. Now, the crux of this whole story is that basically a single point mutation, one change in the DNA sequence, allows this totally different conformation to arise. And so in doing so, it basically allows the alpha subunit to evolve very, very rapidly. So we typically think of evolution occurring in this very gradual process, and in many cases it does. This is actually an example where it occurred very, very rapidly because all of these other cleavage sites existed. It's just that only under a very specific set of circumstances, it actually became selectively advantageous. So again, the important point to remember here, a very simple change in the sequence, and we gained a totally different function. And it was such an important function that it actually has whole venom phenotypes shaped around the function of this pair of neurotoxins, which is pretty crazy to think about. And this whole process actually demonstrates a really important evolutionary process that's called exaptation. And this is similar to adaptation, but in adaptation, we think of phenotypes being optimized for a specific selection pressure. You can think about it being optimized for a specific ecological scenario. Exaptation occurs when you have a trait that gains a totally new function that is independent of the original selective pressures that gave rise to it. So for example, bird feathers likely originally arose to help with things like thermal regulation. But then when birds started to fly, it provided a means of giving lift. And so this is kind of that same idea. It's also called co-opted adaptation, which in some ways is more intuitive because it basically is when you're taking something that was made for one thing and using it for another. And in some cases, using it very, very effectively. And the reason that this is such a important evolutionary concept is because it's a mechanism that may allow traits to bypass what we call low fitness intermediates. So this is the idea, instead of having these gradual changes where you think, well, how is this possible? Because for it to be any other way, you actually have an ineffective phenotype. In fact, you may not have to go through that fitness space at all. You can just bounce between what are called fitness peaks. So this is I know this is a really complex process, but I always thought this was just super cool and a really neat example of evolution occurring in real time in a way that makes a lot of sense and explains some pretty unlikely observations that we see. Questions on this? Because it's a lot to throw out. Yes, Ethan.
So the alpha protein,
Mhm.
the alpha subunit, the
Yep.
acidic one, was pre-existing in the toxic compound before an original point mutation.
An ancestor of it was.
Okay.
Right.
And that original point mutation was to the alpha, and then that allowed it to bind to the beta.
Yep, that's exactly right. Yes, SK. Go for it. Mhm. We don't know exactly what it was used for, but I probably could find out with just a little bit of work. So actually this phylogeny I showed is the phylogeny of these PLA2s. And this ancestor, it's called ancestor CA1, is actually a functional enzyme. So it has its own toxic function, but it cannot bind to the beta subunit. that makes any sense. So it's basically this point mutation causes the loss of the toxic function, but in doing so, it allows it to bind to the beta subunit. That's why we say we're going like from fitness peaks, because if it has toxic function, it's effective. If it can bind to the beta subunit, it's also effective. It's just that if you had that scenario where you don't have either, you're in what's called a fitness valley. It's not effective. Do you have another question, or was that it? Yep.
So the
Mm-hmm. Oh, yes. So the pythons is probably different. So we use the python here as an outgroup because they have their own phospholipase. So it's a little bit different here because even though this is basically the ancestor of all these toxin genes, it may or may not have toxic function, and it probably actually has function as like a digestive protein. This actually kind of links back to what we talked about last week with the Toxicophora. This is an example of a python, which we consider at least functionally non-venomous, having an ancestral toxin gene that probably serves a different function. Yes, Sydney.
I was just gonna ask, do you have any other good examples of
Acceptation.
acceptation?
Well, we have lots of examples of acceptation occurring. And I can't think of any off the top of my head, actually. I know of this one in Venom because I think it's really neat, but there are actually many cases where we believe acceptation has likely occurred. If you just do a quick like Google Scholar check, you'll find lots of examples. And it is this really important, we believe, or many believe, it is an important mechanism of evolution. Yeah. No, go for it.
How new is the term, I guess?
I think it was Stephen Jay Gould in the 1950s that put the term out there. So it's not, I mean, not that new, or I guess depending on how you look at it, it is very new, because that 50 years ago is actually not that long ago. I guess 70 years ago. Ethan, you had another question?
Yes.
Yeah.
I was just a bit confused by the origin
Sure.
of the alpha. So was the alpha originally an exact duplicate of the beta, or a different gene entirely?
So I would say that the beta and alpha have both diverged from a common ancestor,
So
basically.
they're just like, it's like they started off at one point and then they went off their own things. Let's say like 2 million years on, they're completely different.
Yep.
And then this one point mutation allowed it to.
Yeah, correct. Yes, McKenna.
How well do we need to know this process?
So the important thing to know is really the idea that it's an example of acceptation, right? I don't think it's going to be necessary. I don't want you guys to know that, it specifically is this point mutation that occurs at this site. I just want you to know the general concept that acceptation, we see examples of it occurring in Venom, and it provides a way for rapid evolution that can avoid these low fitness intermediates, right? And that sequence evolution is this really key component for phenotypic evolution and for evolution in Venoms. Those are the big takeaways. Okay, so with that, I'm going to talk just a little bit about, oops, I'm bouncing through things. The last thing I want to touch on really is snakebite and snakebite treatment, because I saw some people were interested in this based on the questions after looking at the poster downstairs. So a few things to mention. One, in general, venomous snakes do not want to bite. And we know this for a number of reasons. I mean, one, we see a lot of defensive behaviors that snakes do to basically prevent or to hopefully avoid biting. So for example, rattlesnakes with their rattle, if they wanted to bite, they wouldn't be rattling to let you know they're there. The whole point is to signal that they could be dangerous if you keep messing with them. Another really common behavior is called gaping. This is actually a general behavior that we see in a lot of species. But in snakes, we also see some cases where, for instance, like the cottonmouth is so named because it has this really white mouth, which has been proposed as a signal, don't mess with me, I can bite. They're going to open their mouth and show their fangs. Hey, I've got these big fangs, I can hurt you. We see a lot of snakes that actually do this. So for instance, the tree vipers that I worked with, they gape because they need their tail to hold on to a tree, but they'll do that same behavior and open up, show you their fangs a little bit. A lot of snakes will also do what are called mock strikes, which is where they'll like kind of pretend to strike, but they're not actually, trying. And then other things like hooding is actually, we see hooding evolve convergently many times. If it's not a full hood, it's a partial hood, they flatten out the neck. So all of these are different ways that snakes will actually try to avoid biting. And if you look at even data on the behavior of snakes, of what will make a snake bite, you actually have to work pretty hard. So this is a case study in some cottonmouths that we're looking at how different behaviors basically caused, different human behaviors, what reactions they cause. So for instance, attempting to escape here is the behavior. I mean, if you stand beside it, if you step on it, most of them tried to escape. Gaping, it's the same. If you stand beside it, step on it, you see a lot of different behaviors invoke gape. What I really want to point out though, is if you get down here to bite, if you stand beside for these cottonmouths, if you stood beside it, which means you're not even stepping on it, you're just like kind of in the same area, none of them bit, not a single one. If you stepped on it, less than 10% bit. Think about that. That is insane. If you stepped on the snake, less than 10%. So that's really low. So basically the biggest proportion of snakes that bit, you had to pick them up. So basically this sends a very particular message. Do not pick up snakes. This is the biggest reason, especially in North America, that people get bitten. And I put this picture up, and this is actually a screen capture because I Googled just snake bite. And I thought this was hilarious because the news story, and I'm sure whoever wrote this was just having a grand old time. The news story, if you can't read it, it says, rattlesnake bites Arizona man in the face, neck after he tries catching it. And this was the picture that was associated with it. Yes, Ethan.
I heard something a while ago. I don't know if you know if it's true or not. I heard that the most common statistics for snake bites are males between the age of 20 and 30.
Yep, that's
who
true.
are drunk on their face and neck.
I don't know about, I think the face and neck is probably not as true, but hands is true. Yeah, so those are actually the big thing for snake bite in the US. Those are the 3 predictors that people often cite, and they are accurate. They're backed up by data. The vast majority of bites are young men between 18 and 25 or something like that. The vast majority of them involve alcohol, something like 40 or 60%. I guess it's not the majority, but it is a huge proportion. And then almost all the bites occur on the hand is basically the truth. Is the hand or somewhere that's not the foot, which indicates that they were trying to do something with the snake. Yes.
So for the gaping, why do they need all that energy to not bite? You know what I mean?
So the gape doesn't require energy, right? It's just basically opening their mouth. I mean, it requires some energy, but it's not a lot of energy.
I thought you meant the gape and open their mouth and kind of.
No, they might do some mock strikes, but those are not very. They're also not energetically taxing. And in fact, all of the defensive behaviors, none of them are what you would consider particularly taxing. So even rattlesnake rattles are actually very specially designed muscles because they do very fast muscle contractions, but they're very weak. And so they don't exert a ton of energy to rattle their rattles. Yes, Sydney.
I'm just kind of confused. What is different about stepping on a snake
Uh-huh.
rather than picking it up from the snake's point of view,
I-
them wanting to fight versus not wanting to fight?
So if you step on a snake, the snake is still on the ground, snake is still in its area. If you pick up a snake, you're actively catching it, right? So you might think about the difference between, this is maybe not a great example, but what would you feel more threatened by if someone came over and just wrapped their arms around you, but you've still got your feet on the ground, you still got some autonomy, versus if 2 people came over and picked you up. Right? It's a very different sensation. And especially for these organisms, they want to be on the ground. Yes, Catherine.
we don't see it yet, so it's a new behavior, make sure that you know it's there.
Absolutely. That's very common. In fact, that's really what rattlesnakes do, right? Is that and they, depending on the rattlesnake, you actually can get different behaviors because some rattlesnakes will not rattle. They would rather basically play the odds and say, I just hope you don't notice me. So a lot of snakes do that. Other times the snakes will, for instance, they'll rattle just to say, hey, I'm here, do not step on me because I don't want to be stepped on, So, I mean, yeah, it's absolutely kind of it changes based on species. It changes based on the snake. Yeah. Oh, a couple other quick things I wanted to mention here because I know this was coming up and we are talking about snake bite. So obviously I'm not mentioning much about Gila monsters, but Gila monsters are kind of the same thing. And it's interesting. A couple of years ago, I looked into a little bit about Gila monster bites because I was very curious about it. Almost all of them occur from people that are handling them. A large proportion occur basically from animals in the pet trade or in captivity. I think at the time I looked it up, and this could be incorrect, or maybe I got it from like a pop sci book, so its accuracy might not be accurate or might not be super high, but I do think it's a really interesting story. I think that there's only a handful of recorded Gila monster bites because they're so rare. One of those, though, is perhaps the most unlucky person in the world, I think, because it was a skydiver. And it was a skydiver who successfully completed their skydive, but the moment that they landed, there was this big gust of wind, and it was so strong that it caught their parachute and took them off their feet and dragged them like several dozen yards. And in doing so, they happened to basically roll over a Gila monster that clamped on. And so that's one of like the very few legitimate, like truly accidental Gila monster bites where it was just like an incredibly unlikely scenario. Another couple other just like random things I'll mention. There are certain snakes that I've encountered that I absolutely think if I ever meet someone who says like, I was bitten by this snake, I will forever, no matter what they say, think it's their own fault. So I showed you guys the Microides, which is one of the species of coral snake, right? So those animals are tiny, for one thing. I mean, we're talking like the size of a very large worm. So they're very small animals. They're super fragile. Their mouths are absolutely very tiny, and they just do not want to bite. I mean, and I cannot stress enough how much they do not want to bite, because we were doing a project trying to get venom out of them, and they just, you got their mouth on the parafilm and everything, and they do not want to do it. So a lot of these snakes are really avoiding it as much as they can. Big rattlesnakes are another one. We've encountered some rattlesnakes that are like the very large, heavy body ones that for whatever reason just don't seem inclined to inject venom. They just don't want to. And so I think that you really have to mess with the snake to get them to bite, which is not to say you should all go out and mess with snakes. It's the opposite. It's that if you encounter snake bite, in the vast majority of cases, especially in the US, especially in kind of North America, it's going to be the fault of the person who's doing the instigating, basically. Okay. Now, despite all of this, a couple other things about this. So this is something that came up, I think, but an important thing to remember, snakes at all ages maintain control over their venom expulsion, so they do not have to inject it even if they bite. Another really important behavior that we see in snakes, and I guess not really in lizards, but definitely in snakes, but also in other venomous taxa, things like scorpions and spiders, is that we basically see what's called venom metering. And that means that the snakes select or choose how much venom to use in any given scenario, which includes dry bites. So when the snake bites and doesn't inject any venom, but also interestingly, when snakes bite prey, for the most part, they don't empty their glands into the prey. And so there's these different ways that they can choose whether or not to inject their venom. Sydney, is that a question hand or just a?
Yeah.
Okay, go for it.
So you know that getting injecting venom into prey is costly.
Mhm.
So then does that mean that it's costly to like milk a snake?
Yes, hypothetically, I believe so. I mean, we imagine that whatever the cost is to the snake injecting venom into a prey item or defensively, it would be the same if a person is milking the snake. Yeah. We give them recovery time. So I'll mention our next, ooh, that was unexpected. So the next slide up here, one of the reasons for expectations that snakes meter is one, we know that venom is not infinite. I think that was a question someone asked earlier, was, can the snakes just produce as much venom as they want and use it as much as they want? And the answer is no. It's not an infinite thing. It's a gland, so they produce it and it fills the gland. And then once the gland is emptied, it is emptied. And it takes about a week to fully replenish the venom gland. So a lot of the people that I know that milk snakes regularly, for instance, there are farms that milk snakes for use in drug development or for anti-venom development. They often go on like a 2 or 3 week cycle for their snakes. So they milk the snake and then it gets 2 or 3 weeks to rest to replenish its glands and basically not be stressed out. Now, interestingly, because this is a question that came up, I specifically looked into this, was what are the energetic costs with venom? Because it is kind of assumed that venom is a costly substance. And it's certainly, at least I was assuming, is a metabolically costly item. But in fact, I think a lot of the recent evidence, which there's not a lot of research done on it, but a lot of the recent evidence that I was able to find actually suggests that the costs, whatever they are not metabolic. That the metabolic cost of producing venom is basically akin to just like standard functions, including growth and things like that. So that's not to say it doesn't have a cost, but it might not be, an energetic cost. It could be something more like, for instance, and this is to explain things like venom metering. It could be things like if you're using your venom and you use all of your venom right now for this prey item, maybe you miss a prey item in a week, or maybe you don't have any venom to use defensively, those sorts of things. It's not entirely clear. But we certainly do know that there is some cost associated with venom because we see instances where the selective pressure to maintain the venom is lost, and we see degradation of the venom, both in terms of the quality of the toxins and of the structures themselves. Yes?
So is that kind of why some snakes are like more venomous for their own good, so they use less venom to like kill prey or for difference, so they have
Yeah. So the question of variation in toxicity is actually pretty hard to answer, and it's not something we're really clear on. A lot of the explanation that I'm aware of that I think are the main arguments are that like very toxic venoms evolve in response to arms races with their prey. So there are cases where it's very toxic to a lab mouse because a lab mouse has not been evolving resistance to that venom for several 100,000 years or millions of years. Whereas if you inject that venom into a local mouse, a prey that snake might actually be eating, it's going to be far more resistant. And we do see lots of cases where, in fact, prey are much more resistant to the venoms of their predators than basically you would expect in other scenarios. But there's also arguments that say we don't really know enough about what is going on in terms of in a lot of those interactions, and that even a lot of those cases might not explain all of the toxicity we see. And I've seen several hypotheses on it that I'm happy to chat with you about at some point, but maybe not at this moment because a lot of it's postulation on my part. Okay, so beyond that, we also, again, we see dry bites. So these are times when the snakes bite and don't inject venom. The statistics on these, I think, vary, but I've typically heard somewhere in the neighborhood of a quarter to a third of all snake bites are considered dry bites where there's no venom injected at all. These don't include colubrid bites. So those are scenarios where, it's maybe there's no reaction or maybe there was no venom. These are specifically cases where it's something like a rattlesnake or a coral snake, a snake that's known to be venomous, and it chooses not to inject venom. Yes, Sydney.
I was just gonna ask, in terms of conservation,
Mhm.
how accessible do you think this knowledge is to the average non-scientific person?
I think it's very accessible if you look for it even in a cursory way. But I think most people do not look for it, right? It's like a lot of knowledge. You know, if you did just a little bit of digging, it's really easy to find, but most people aren't interested. Yes, SK. No. A dry bite, I mean, if it were fatal, it would be, I shouldn't say no, because of course there could always be some sort of crazy circumstance or exception, but by and large we expect no. It would have to be, I don't even know of a scenario, but you know, where it like it'd be some other health complication that would drive it. So I guess hypothetically, here's a scenario. This is actually a very realistic scenario, especially many years ago. If, for instance, you received a dry bite, but you treated it in an extreme way, like for instance, removing that appendage, which is a thing that people really would do, yeah, that could be fatal. And you could argue that that's a consequence of the dry bite, but you know, that's not really a consequence of the dry bite. Okay. So despite all of this and all the information that I've just told you about how unlikely and improbable snakebite is, especially to us, globally, snakebite is actually still a big problem. And the big thing is it doesn't affect everyone in the world equally. You'll notice many countries that have a much bigger problem with snakebite than we do in the US. So in the US, we're looking at less than 10 deaths a year. But in other areas of the world, you could be looking at over 1,000 deaths, or in some cases, over 10,000 deaths. And one of the big problems and one of the reasons that it affects different parts of the globe differently is that snakebite is disproportionately a problem for those in rural areas and those that are in impoverished communities. And this is for a few reasons. One is that it has to do with your likelihood of encountering it, but probably the bigger reason is that it has to do with your probability of being able to receive appropriate medical attention in a timely way. And so even in these cases where we're looking at these like global trends and we see huge amounts of snakebite, in fact, the vast majority of snake bites occur from just a handful of species. So see, these are some of the big players that are known throughout the world. So in the top left, and you guys don't need to know what species these are, but I'm just going to give some examples. This is the saw-scaled viper. On the right is the puff adder. This is the cobra, I'm guessing the Indian cobra. The bottom left is the commoner banded crate. This is a Russell's viper. This is the jararaca. This is it is the fertile lance, but the fertile lance is a common name that's actually applied to all lance heads. So this, you can really only refer to it by its scientific name, which is Bothrops asper, but it is commonly called the fertile lance. And then last here we have the taipan. And so these are, you know, India, Africa, India, and when I say India, it's all of really South Asia. Brazil, these guys are a huge issue in South America and Brazil. This is Central America and also Northwestern South America and Australia. So these species cause the vast majority of bites, and they have many cases, they have very potent venom, which is why they're especially problematic. So the biggest and best way to treat snakebite, at least at the moment, is with antivenom. So antivenom is a specialized protein solution, and it is basically contains protein products that bind and neutralize the venom proteins. Now, importantly here, these venom or these antivenoms do not undo any damage that the venom does, because once it's done, it is done. You know, there's not a lot you can do if the venom has basically deteriorated a lot of the muscle tissue in your arm. You know, the antivenom can't replenish that. That's natural healing. But what it can do is bind these proteins and stop them from doing their function. And so there's this saying in relation to snake bite, which is time is tissue, which means that once you've been bitten, the more quickly you can get medical attention, the more quickly you can receive access to antivenom, the better off you will be, the lower its effects are. Now, all antivenoms that we're currently using, to my knowledge, are still actually derived from natural immunoproteins, and these are harvested from other animals. So specifically horses and sheep, I think, are the two big ones. And different countries and things actually use different animals. So for instance, this is crowfab, which is what's produced and you're allowed to use in the United States. It's actually not a very good antivenom. It's produced from sheep. And in Costa Rica, at least, the big producer of antivenom is an institute called the Instituto Clodomiro Picado, and they use horses. So they have this big farm and they have a ton of horses, and the horses get regular injections of these different venoms. And then they harvest the horse's blood, spin it down, extract the serum, and they'll take these immunoproteins from the horses, do a little bit of processing, and then that's what you get when you receive an antivenom injection. Yes, Sid.
I don't remember which group it was, and I don't remember the species of the snake.
Mhm.
But somebody said that there's no antivenom for a certain species.
Mhm.
What makes you not be able to get antivenom? Is it just that it hasn't been studied, or that there's not a way to synthesize it?
Well, so there's a number of things. Some of these I'm going to mention now. So one of the things that's important to know is that antivenoms are basically limited to the species venom that are used to make them. So if you think about the way that this is working, the horses or sheep are only going to make antibodies that can bind or inhibitors that can bind the proteins that they're being exposed to. So they're not going to make an inhibitor or something that can affect the venom it's not being exposed to. And so some venoms are made using multiple species, and other venoms are species specific. So one of the things that can actually affect whether or not you have an antivenom is basically on how available those species are and how much venom they produce. So some species don't produce a lot of venom and they don't cause a lot of bites. And to be very honest, it's just not fiscally, there's no fiscal incentive to produce an antivenom. You know, if you're a snake and you're producing, or for instance, a species of snake that's causing 100,000 bites a year, yeah, you want an antivenom for that. But a species of snake that might be just as deadly, but is maybe only biting 10 people a year, the cost just might not justify it. Yes, Athena.
So the venom that you have to inject in the sheep or horse, is it the same, like what would be a lethal dose for human, or is it like
Higher, usually. They make it so that it would be typically more than what it would be for a human, but it's not so much that it would like extremely tax the horse. So I've seen actually there's some argument that this is like an inhumane thing to do because you're like, you know, hurting the animals. I think that in many more cases, it's actually not that the venom is doing, I mean, the venom is going to be problematic for the horses. It probably is going to make them not feel well for a few days. But I suspect that in many cases, what's more important is the condition the animals are being kept in. Because if they're well fed and their immune system is up, then the doses of venom they receive should not be problematic, if that makes any sense. So in general, a couple things to note, this process is very inefficient. And it's very expensive because you're basically making sure that you maintain these animals, which is very expensive. And then all the processing is very expensive. There are also issues because these are natural proteins. And even though many of them are processed to make them basically more effective or give them a longer shelf life, they do still expire pretty quick. And so because of that, that means that they're expensive and they expire quick, which is what limits a lot of people's access to them. And I said that a lot of what contributes to snake bite issues is that people can't get to antivenom. Well, that's in part a function because they're so expensive that a lot of places don't have them. And you may be frankly too far away for a hospital to actively maintain a stock. Now, all of that said, there's always ongoing research into other ways to treat snake bites. I am not super well read on that subject, but I've seen a couple of presentations about different approaches. I know that there are groups that are working on actually, for instance, certain types of medicine that tend to treat the symptoms and seem to stop snake bite for a certain amount of time. So it would be the kind of thing where you might be in Africa and you are bitten by a venomous snake and you pop a pill really quick and it buys you about 4 hours. And then that'll give you the time to get to a hospital that has antivenom. There are also other types of antivenom that they're working on making that are a little more robust, a little less prone to being to like expiring and things like that. Yes, Cindy. In what way?
Like, I remember I don't remember, I think it was here, so they gave about the environment and evolution of it.
Uh-huh.
They were talking about like lethal dosage and how that was actually measured.
Okay, so you're talking about lethal dose as a measurement.
Yeah.
Yeah, so there's this term that's called LD50, which is what you often see referred to as the lethal dose. It's actually the mean lethal dose. And basically what that measure is the amount of a substance, that it can be venom or anything, at which 50% of a population is expected to expire when they receive it. So for instance, to calculate a lot of what we see is listed as like the toxicity of venoms, what they do is you would inject mice with different amounts, and you might inject, I think it's typically done at like 4 levels. So you do like 3 mice with this amount of venom and 3 mice at this amount of venom and 3 mice at this amount of venom and 3 mice at this other, and you produce a curve. And based on that, you can estimate at what level 50% of the population would expire. So that's what the lethal dosage is. That's how it's calculated, basically. Okay, so that's all the info I have on antivenoms, and that's actually all the info that I've got for you guys total. So this slide, just really quick, is just takeaways, some big things that I want you guys that I've, if you don't remember anything else, which I would recommend remembering more than these points, but if you don't remember anything else, at least remember these things. So we know that venoms are these really important evolutionary innovations. We talked about some of the different ways that they play a key role in the lives of these species, of venomous species. Diet is the key selective pressure. We saw several examples of evidence of that. We also looked at some examples of venom variation, and we talked in great detail about the genetic mechanisms that underlie them. And then we also looked at some differences in the venom delivery systems among lineages. We know that they're highly variable. And in fact, the origins of venom systems, broadly speaking, is still somewhat contentious. And with that, I'll stay on this slide for a minute, but I'm going to open the floor to questions with a couple other exceptions. So if you guys want to start pitching questions, you're welcome to. Happy to answer questions about anything at all. Yes, Dr. Ingloff.
We can do the survey that we had in class on Wednesday.
Oh, okay.
We're out of time now.
Oh, are we?
Oh, yeah.
Oh, okay.
So if it works for
Sure.
you,
Yeah.
we can make class time on Wednesday to do that.
Yes.
And just want to make sure we do get to the announcement slide.
I was about to say, yeah. So Sydney, I'll come right back to this, but I want to show this for everyone. So, yes.
So we rescheduled this one trip. It's going to be Monday during class and in the evening. So we're going to do both trips on the same day. Just a reminder, you're required to go to one or the other. I have a survey for you in your program for Wednesday that asks you what you're doing for each of those trips, where if you're going, if you're going to drive yourself, that way we can plan accordingly. So please, everybody, make sure you do that for Wednesday because we'll need that information to plan this out. So yeah, that's good news. We're going to go see mud puppies. The weather looks great. Another announcement, your quiz is Wednesday, and it's going to be, I'm going to change one thing from what I previously said. It is going to be open book. So it covers a lot of material. See previous slides for what we're covering in your quiz, but instead of closed book, it will be open book. We don't have time for a review session, and there's a lot of material, so I think that'll make things a little bit easier for you. Yep. You can use it, yeah, if you want to bring, I'm not going to monitor that. So, I mean, because I have no good way to distinguish if you're looking something up on the internet and versus your own notes. I will say this though, with open book stuff, I highly recommend using your notes from class or other references provided in class and not just trying to Google answers, because you'll get some wackadoodle answers that won't make any sense and will be completely irrelevant to what we covered in class. I see this very often with students with open book stuff. So you can use whatever you want. I'm not going to monitor it, but my recommendation is to use material from class because that will give you the answers that I'm looking for. Yeah, Sam. Yeah, I'm still going to do the same amount of time. Yep. So with that in mind, you won't have time to look up every answer from scratch. So be prepared to take the quiz as if it was an open book, but know that you'll have a backup and some extra resources. So organize your notes, etc. Thanks for asking that. And with that in mind, after spring break, when we get back, our first class back after spring break is going to be a review session. We've covered a lot in the first half of the semester. We're going to take a whole class period to review it, organize it, get your brains back out of spring break mode into herpetology mode, which, you know, for some of us are synonymous, but whatever. And then we have your first exam is going to be that Wednesday. I'll provide some details coming up, but I just wanted to get that on your schedule. I toyed with the idea of having it next week, right before spring break, but I didn't think that would be great. I know a lot of classes are giving exams that week. It felt a little rushed to me. So we're going to do it after spring break and take that whole first class as a review session. So let me know if we're out of time now, but just let me know what questions you have. We can talk about this more in detail on Wednesday. Otherwise, do you have anything else to add? Nope. Okay, let's give it up for Dr. Mason and coming in and treating us with an awesome meeting on Venom. We are out of time, but thank you, everybody, and I will see you on Wednesday. Be sure to see your class program for Wednesday. There's 2 short things on there for you.
I just wanted to tell you that I have my quiz scheduled with the testing center on Wednesday
Perfect.
at like
1:40. Okay, that should be perfect. Then you can take it
Yeah,
and then just come here
exactly,
afterwards.
and then
Yeah,
come straight here.
that's completely ideal.
So
Yep.
I did that and I didn't put in that it was open note or open book. But I
Okay.
plan on emailing them, but they might.
I will, when I send them the exam or the quiz rather, I will make sure they know it's open.
Okay,
Yep, yep. So
Thank
it should be
you.
good. Yep, thanks for letting me know. Perfect, same time even?
Mine's gonna be at noon.
Okay.
It's just when I filled out the thing, I forgot to put that it's open out.
Okay.
Instead of the thing.
You didn't know it was open note. Yep, I will make sure they know. Yep, yep, thanks, Tom.
Because I wouldn't know. I didn't know about the issue, but if I did, then
Yeah.
I'll still
They would just